all. This is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. So, the discussion today is about a drug called Tempol. It is by Adamis Pharmaceutical. So, let me very quickly start. They have requested a fast track, they have submitted a fast track application to FDA for COVID 19 management at home. This would be one of the first drugs that may be, if approved or authorized, that may have been useful at home. So let's see this. I think this is a unique drug. So let's look at the references. So this is drbean.com. This is Adamis Pharmaceuticals, and this is the news that they submitted fast track application to FDA for Tempol for the treatment and prevention of COVID-19. And I want to give credit to Gene Bean and John Snyder on Discord to post this information. So here is that information. We'll continue in a second. This is the for the Tempol and COVID-19. There was a clinical trial that has been going on, and so I think that trial is getting ready for the publication. So if you see here, Tempol, a novel antioxidant. So keep these three in mind, and I'm going to explain them. Antioxidant inhibits both activated T cells and antigen presenting cells. I'll talk about that too. And then inhibits the APC derived cytokine storm or cytokines in vitro for COVID-19 patients. So in that study, they say that pre-incubation of immune cells, and I'll explain this, I'm just kind of showing you a summary of what is happening. Pre-incubation of the immune cells with Tempol resulted in a significant, and P is lesser than 0.05, so significant, decrease in multiple T cell and antigen presenting cell derived cytokines from both cells of COVID-19 and uninfected individuals. These results suggest that Tempol has strong in vitro, so this is in vitro, cytokine activity and supports additional studies examining the use of Tempol for the treatment. Tempol has strong, broad in vitro anti-cytokine activity. Suppression of inflammatory cytokines with an antioxidant may be beneficial strategy for COVID-19. That was their background. If you look at this more data over here, the study that they have, in that study, they will be giving Tempol capsule. And by the way, Adamis Pharmaceutical has the worldwide license for the use of Tempol for the treatment of respiratory disease, including asthma, respiratory syncytial virus, influenza, and COVID-19. So the trial, which has 124 people, they would receive 800 milligram 400 milligram twice daily, total of 800 milligram, up to 14 days. The results of a published study in collaboration with Stanford University researchers suggesting that Tempol has strong, broad in vitro anti-cytokine activity. Suppression of inflammatory cytokines with an antioxidant may be beneficial treatment. So the only two words I would request you to keep in mind as we go through this discussion are, number one, it is antioxidant, and number two, it is anti-cytokine. Now, in terms of anti-cytokine, we know many drugs. For example, we know leronlimab blocks one part, or Rantes. We know, for example, Miraviroc does that. We know steroids do that. We know many other anti-inflammatory drugs. So what is the difference of these drugs from the other, or these drugs from each other? That is at how many cytokines and what kind of cytokines they suppress. So you would be amazed at the cytokines that this drug suppresses. It suppresses a set of cytokines, not all, a set, which is a very interesting set for inflammatory outcomes. So we'll look into that set in a second. So that's the basic difference. Plus, this is an antioxidant. So a drug that is antioxidant, so generally beneficial for inflammatory states, plus anti-inflammatory, and that is selective anti-inflammatory, is a very useful drug. That is the basic idea. Then they say the Tempol has already been shown to be well tolerated in human clinical studies to date. Good. Now, I have no disclosures. I have no financial, commercial, any kind of, affiliations with Adamis or any strings attached. This is the NIH. So, and mind you that NIH actually has an article on ivermectin as well. This is NIH saying Tempol, a potential home treatment for COVID-19. So in the case of mechanism, antioxidant and anti-cytokine, in case of usage, at home treatment. So look at what NIH is saying. They are saying Tempol that can be taken orally and may stop the replication of SARS-CoV-2, the virus that causes COVID-19. This treatment would likely prevent severe disease. And just like the other drugs that you are aware of, for example, IV, this drug also doesn't care for the variant. It would just act on the immune system and prevent the cytokine storms potentially, reducing the severity of the disease or potential for severe disease, regardless of what variant it is. So let's see what NIH says further. SARS-CoV-2, so this is a very interesting statement. SARS-CoV-2 needs clusters of iron and sulfur. So most of the pathogen, when they're replicating, they need raw material. A lot of pathogens need iron for their activities or replication. So here they need iron and sulfur. Many of those iron and sulfur minerals or ions, they go in building blocks for genetic material, but also for the other enzymes as well. So iron and sulfur minerals to replicate in cells. Tempol breaks up these clusters and stops the virus from multiplying. Isn't that interesting? This action might prevent severe COVID-19. So what are the mechanism of actions that NIH thinks are interesting? Number one, calming of inflammation. SARS-CoV-2 infection sometimes causes an out-of-control immune response called a cytokine storm, which can damage the lungs and other organs and may cause death. Tempol may help prevent cytokine storms. Tempol's antioxidant activity may protect cells by inactivating free radicals. This is just like vitamin C or even other vitamins. Unstable molecules made by out-of-control immune response that can damage organs. So free radicals, especially the nitrogenous products, is what Tempol works on. These products, when they are free radicals, they can attack the cell's own machinery and damage them. Imagine these are like tiny bombs that are going off inside the cell. So whatever enzyme or protein they go off near, that would be damaged. And sometimes they go off near the cell membranes and they fuse the cell membrane, they cause holes in the cell membrane, they create punctures in the cell, they can even end up causing the cell death. Then you would like this one, the last one, decreasing clumps of platelets. See, COVID has a problem with clotting. Tempol is anti-clotting as well. So it is antioxidant that is needed, anti-cytokine that is needed, and antiplatelet. So platelets, so really not antiplatelet, but meaning it reduces the thrombosis or platelet aggregations. Platelets are blood cells that help stop bleeding. If they are too sticky and form clumps in blood, meaning they become thrombi, as can happen in people with COVID-19, dangerous blood clots can result. Tempol may reduce this risk. Can you imagine? Almost every single pathology or the baseline mechanism of pathology can be attacked and addressed by Tempol. So then they say, why is this research important? Because they're asking for a research on this one. The US Food and Drug Administration has given the go-ahead for a study of Tempol in humans to see where it can safely prevent hospitalization. So that is the NIH. Then this is the study So here, brief report. Tempol, a novel antioxidant, inhibits both activated T cell and antigen presenting cell derived cytokines in vitro. This is what I'm going to explain to you. So if you just wanted to hear the summary of what it is, at home drug, 400 milligram capsule, 2 capsule per day, 14 days at home, and hopefully it would keep the symptoms and the cytokine status in check, that cytokine storm doesn't occur, severe disease does not occur. Number one. Number two, you'll see further, it becomes active when the cells are active. It does not suppress resting cells. That means if there is a problem for which a resting cell needs to function, Tempol will not suppress that cell. It would let it become active. Those that are already active and are producing cytokines, it blocks them. So active war that is going on is what it would attack. That is very interesting. So in this study, we developed an in vitro assay. So this is an in vitro study using activated peripheral blood mononuclear sites. So now, peripheral blood mononuclear cells. So let me quickly show you my diagram. This is my diagram. So the peripheral blood mononuclear cells are the cells that have a rounded single nucleus. In the blood, there are cells that have rounded single nucleus, for example, B cells, T cells, natural killer cells, monocytes, and so on, mostly the immune system cells. There are cells that do not have a nucleus, for example, platelets and RBC. Then there are cells that have multi-lobed nuclei. So this drug works with the mononuclear cells or peripheral blood mononuclear cells, monocytes, dendritic cells, macrophages. Dendritic cells are normally in the tissues. Macrophages, the T cells, B cells, and so on, natural killer cells. That is one. Secondly, if you see here, if they're stimulated with lipopolysaccharides, so what happens is lipopolysaccharide is a very classic stimulant for drugs. So those who were looking for a summary, we're done with the summary. This is how it helps. It would be at home drug. Now we're going in the detail. So many times bacteria have lipopolysaccharides in their walls. And these are really irritant for our immune system. They really bug our immune system. Our immune system reacts to them. So many times when researchers want to irritate an immune system cell, they expose that cell to LPS, lipopolysaccharides. So imagine here on this left side, here, this is a, let's say, dendritic cell. If the researcher want to see or activate this dendritic cell, they can put a lipopolysaccharide molecule next to it, or not one molecule, a bunch of molecules. And when this macrophage will connect with these lipopolysaccharides, it will become active. And when it will become active, I think you know that this is the innate arm. So it would start producing various interleukins. It would then connect with the naive cell if it has some antigen with it. Then the whole adaptive arm will become active as well. So how do you artificially activate these cells? You show them lipopolysaccharides. That is one. Or how do you activate the T cells? You don't show them lipopolysaccharides to activate them. To activate them, you can show them CD3 plus CD28 molecules. So in general, you irritate the immune cells. They become active. They start releasing cytokines. Then you put some drug on them to see if they would stop releasing them. Or you already treat a cell, you already pour on its head the drug, and then you send that cell in the war zone and see what does it do over there. And if it doesn't do anything, that means that drug has inhibited that cell. This is what they did. They pre-incubated the cells with Tempol. So they treated them. Then they brought those cells in the field, in the war zone, and expose them to the lipopolysaccharides and to CD3 and CD28. And then they wanted to see if these cells are going to produce cytokines. And they saw that they did not produce all cytokines. They produced all, but they did not produce an important subset. So that's what we'll talk about. So they say that we hypothesized that a novel antioxidant called Tempol may decrease cytokine from activated peripheral blood cells from both COVID-19 and normal donors. So they wanted to see that this drug would do it for COVID-19 and others as well. What does that mean? So let's say if I don't have COVID-19 and you're giving me this drug, then hopefully you are kind of prophylaxing me by keeping my cells in control, that if they become exposed, they will not go mad. So if this becomes authorized, there is a potential for authorization for prophylaxis and for at-home management of acute disease. So check this out, the green part. Pre-incubation of immune cells with Tempol resulted in a significant decrease in multiple T cell and APC derived cytokines. So I'm now going to go to the cytokines themselves. We'll look at this later on. So let's go and check the cytokines. So, excuse me. These were all the cytokines that they were testing for. And here is the list of cytokines that got blocked. So I'm going to very quickly talk about these cytokines that got blocked by Tempol and what is the importance of them. So if you see here, interleukin-1 beta, granulocyte monocyte colony stimulating factor, macrophage inflammatory protein 3 alpha, interleukin 10, 2, 6, and 21. Let's look at that in our diagram. So here, imagine you have activated a macrophage. When the macrophage is activated, remember it, we have done this discussion, it produces interleukin 1 and interleukin 6 and 8 and GM-CSF and 6 and so many more, even interleukin 2. So out of the whole set, interleukin 1, which is very important, remember interleukin 1 goes to the liver, then it goes to the hypothalamus as well. It resets the temperature centers, it causes fever, it triggers the acute phase proteins, or kind of helps trigger the immediate reaction. Then granulocyte monocyte colony stimulating factor means that it is a chemical substance that is released by the dendritic cell monocyte macrophages. This goes to bone marrow and tells the bone marrow to make more granulocytes. Granulocytes are also part of the blood, for example, eosinophils, basophils, neutrophils, more immune cells, plus monocytes. Monocytes are the cells that are going to become macrophages. So macrophage is saying, I need more of my type. So you can see that with these interleukins and cytokines, more inflammation will occur. And if we suppress them, then more inflammation would not occur. Similarly, interleukin 6 is released as well. That also goes and work on the liver. And we have seen this discussion before as well, that when the macrophage connects with the naive T cell, the naive T cell in the presence of interleukin 4, in the absence of interleukin 12, will become T helper 2. T helper 2 will release a bunch of cytokines as well. And out of those, interleukin 4, interleukin 5 are important. They would help B cells to become activated and become plasma cells. Plasma cells will make antibodies, and the humoral response would come together. At the same time, we also know that interleukin 10 is produced. Interleukin 10, remember in our past discussion, one function of that is to suppress the immune system, not forever, for the time being. And the second function of interleukin 10 is to do more chemotaxis and more inflammation and more immune cells coming in this area. So the interleukin 10 is also suppressed. I have written the ones that are suppressed over here. Then interleukin 2. Interleukin 2 is in the T helper 1 pathway. Remember that naive T cell would become T helper 1 in the presence of interleukin 12. Then the T helper 1 will produce interleukin 2, which would activate a CD8 or cytotoxic T cell. Cytotoxic T cells in turn can also produce interleukin 2. They are also pro-inflammatory. They are also cytokines. Then interleukin 21 takes part in T regulatory system. It also is released by follicular T cells. Follicular T cells are the T cells. T cells are helper T cells and the cytotoxic T cell. The ones that live in the lymph nodes are called follicular T cells. There are follicular B cells as well. Interleukin 21 acts to activate them as well. Interleukin 21 also activates natural killer cells. Remember, natural killer cells are part of the innate arm as well. And I always make them with an eye patch. They're like really tough guys. And why are they tough guys and badasses? Because they would touch every other cell. I also call them pervert because they would touch every other cell. They would look at the density of the clusters of designations of on various cells. So if you remember, I say that they normally check the hair present on other cells. And if the cell is sick because of a virus or a cancer, the hair on the cell surface are gone or reduced, and natural killer cell can figure that out and attack that cell and kill it. So interleukin 21 helps with that as well. So a very selective subset. So you could have a valid question here to say, why not we just steroids? Yeah, sure, we can give steroids, and in many cases, steroid would work in later stage when the patient is in cytokine storm. But in the early acute phase or in the prophylaxis phase, you are not going to give steroids. In the prophylaxis, giving steroid is just very dangerous because steroid would suppress the immune system, and now patient is open to many other issues, plus the side effects of the steroid, plus open to the infections. So you don't want to do that. Even in the acute disease early part, giving steroids, mostly, at least here in the US, we know there was a study that showed that in the presence of steroids early on, the coronaviruses becomes difficult to clear them. So you don't want to give steroid even in the acute phase early part. Then you can have drugs like maravirox or leronlimab or other such drugs that would selectively block various chemokines or cytokines. Here you have a drug, Tempol, which is actually, it blocks a bigger subset of interleukins. So it actually keeps, plus it does not attack or suppress the resting cells. So if the body needs those cells to become active, they can. It actually only causes modulation of the active cell. So even within the active cell, it does not stop them. For example, macrophage produces a bunch of other interleukins as well. It doesn't stop them. It stops a select number of interleukins. So it kind of modulates the immune system's response towards a smaller response, manageable response, more beneficial response for the body, and lets it not get out of hand in the form of cytokine storm. Beautiful drug. I love it. And then the idea of this being at home is even more fun. So here, if you see, they said cytokines for which at least 2 concentrations of Tempol were significantly decreased, from no Tempol included, interleukin 1 beta, GM-CSF, MIP3A, IL-10, 2, 6, and 21. They said it did not bother with the other cytokines, which is good, plus it did not bother with the resting cell, which is also good. So it's a beautiful drug. Okay, continuing. Here is a study. This is one of the studies. Then this is just some more references. What are the peripheral blood mononuclear sites? What is the interleukin 1 beta, GM-CSF, and so on. So this is where I will stop this talk. I am pretty pumped about this drug. What I do not know is, number one, will they actually get the authorization? We have seen that FDA kind of is not very much in favor of drugs which are not coming from certain bigger pharma, maybe. But who knows? This has some Stanford studies backing as well. So this is one. Secondly, I do not know the pricing. So I have no idea that if it is authorized, what would be the price. And I'll close with this, that once again, antioxidant, anti-inflammatory, anti-clotting, all three in one. And selective anti-inflammatory, not general anti-inflammatory. So all of those are great things. So this is the discussion. Thank you very much for being here. If you would like to support this work, please like, subscribe, and share. That is the smallest thing you can do. If you would want to do something more, there are 3 links in the description. You can buy me a coffee, or you can use PayPal, or you can be a patron. Thank you very much, and I'll see you in another few minutes.